What you have seen until now, yesterday and this morning, is a little bit the workflow or how we identify proteins in mass spectrometry. Okay, you have seen a little bit of sample preparation, manual interpretation of the spectra, and also Christian just explained automatic database search. Now I'm going to add another level of complexity, which is post-translational modifications that make things a little bit more complex, or quite a bit. So just as in, the complexity added when we go from genes to proteins, in which the numbers increase because, among others, we have alternative splicing, proteins get chopped, etc. When we take into account post-translational modifications, these numbers go now to millions. So the number of analytes that we have in our samples now increase. And it's not only that, but each one of these modifications that are, adding things to our proteins have different masses, behave differently. Usually they are in very low amount. And on top of that, we cannot just take our sample and say, okay, now I want to see all of my modifications. No, that doesn't work like that. So the good news is that each modification adds a mass shift or some change in the mass, and therefore this is a problem that we can approach with mass spectrometry because we can measure these masses. Because this field is quite wide, we're going to focus only on these modifications that you have in the slide. I'm going to move this. And if you have other questions about other modifications, we can discuss also later. So we are going to start with phosphorylation because it's one of the most widely studied and that it has a lot of interest in the community. And this is more or less the organization that we're going to do for each one of these modifications. Phosphorylation, I'm going to explain more extensively, because some problems, we see in common with other modifications, like why we need to enrich or site localization. This is something that happens with each modification. And we're going to go through a little bit, so how is the chemistry, what is this modification, how do we enrich, which strategies we use in mass spectrometry, how do we localize it, and talk about some real examples. Okay, so phosphorylation has drawn a lot of attention because it's responsible for a lot of the communication and cellular processes. Okay, so cell signaling. Many times, due to phosphorylation, processes are turned on or off inside the cell. Kinases are the proteins that add the phosphate group in the protein, and phosphatases remove it. And the amino acids in the peptide that get phosphorylated are serine, threonine, or tyrosine. And not everything gets phosphorylated, but kinases are pretty specific, and they have some specific substrates or specific motifs that are their favorite ones to phosphorylate. Okay, so this is how phosphopeptide looks. This is phosphoserine. This could be the protein or a peptide. The serine, in which the hydroxide group gets phosphorylated. This is the phosphate group. Same with the phosphothreonine and the phosphotyrosine. Something we need to bear in mind when we have a sample with a phosphorylation and we want to look for phosphorylation is the chemistry of it. So in general, you have seen that in our sample preparation, we are working in acidic conditions because we want our peptides to be ionized when we put them in the mass spectrometer. And we work with peptides positively charged. That's why we work in acidic conditions. In these conditions, when we add a phosphate group, this one adds a negative charge. So this would be a typical peptide with two positive charges. In the lysine or arginine, assuming that we are working with tryptic peptides, we will always have a positive charge here. and another one in the N-terminus. Here is a glutamic acid, for example, that doesn't have any charge, in principle, in these pH conditions. And when this serine gets phosphorylated, the total charge of the peptide gets reduced by 1. Okay? This sometimes helps us or not, but we need to know this. As I was telling you, it's important to enrich our sample for these phosphopeptides. Most of the time we are working with whole cell lysates or pretty complex samples, and we know that the phosphopeptides in our sample are, in very low abundance. And it's not only that, it's that phosphopeptides have a less efficient ionization than the non-phosphorylated counterpart. Therefore, if we want to see them, we need to do our best. If our sample is quite simple, it's only a few proteins, maybe we don't need to do this enrichment, because with the instruments that we have now, they are, good enough to see the phosphorylations, but most of the time it's important to enrich. So there are 3 main ways that we enrich for phosphopeptides. We can enrich at the protein level or at the peptide level, but these are the 3 main ways. Either using antibodies, that is used mostly when we work with phosphotyrosine, by metal affinity, that one is one of the most common ways of enriching, or is also been done by chromatography, like strong cation exchange, taking advantage of this difference in charge of the phosphorylated peptides. The enrichment with antibodies is done mostly or all the time with phosphotyrosine, because phosphotyrosine is even less abundant than phosphoserine and phosphorthreonine. There are less tyrosines that get phosphorylated, and therefore it's very important to enrich for them. The good thing is that there are antibodies that can attach to phosphotyrosine, which doesn't happen in the case of serine or threonine. And this can be done at the peptide level or at the protein level. The group of Forrest White, for those of you that are interested, has published a lot of papers related with phosphotyrosine in cancer and other things. And also, most of the time, not only we enrich with using the antibodies, but we also combine this with the other strategies, even if we are looking for tyrosines, because, it helps, but it's not perfect. The most common way of enriching for phosphopeptides is by metal affinity chromatography, which is based in the principle that this phosphate group with the negative charge gets attracted by the positive group of the metals. Different metals have been used. The most common one is iron. And then, in general, these metals are attached to some beads. And by chromatography, we can enrich for these phosphopeptides. One thing that happens is that when we have acidic amino acids in our peptides, can also be a little bit enriched, or at the same time, if our peptide is very basic, meaning has lots of positive charges, maybe it's going to be harder to enrich, because it's all based on charge attraction. And in general, it's easier to enrich for multiphosphorylated peptides. So the 2 most famous beads to enrich for phosphopeptides that I'm sure you have heard of is IMAC, and the other one is titanium dioxide. There have been many different protocols published for this titanium dioxide and IMAC. Every lab has its own little tricks and protocols, but all this is published and it works. Okay, the selectivity is increased when we work with very high acidic conditions. Again, there is a higher affinity for multiphosphorylated peptides, and sometimes peptides with multiple basic residues, we find that they have poor binding. Okay, so just to sum up, these are the two mainly used ways of enriching for phosphopeptides, which is basically a positive charge chromatography that binds to negatively charged phosphate groups. This is the pH that we usually work with, and at this pH, this is more or less the pK of the phosphate group, the glutamic acid and the aspartic. Of course, these pKs change depending of the neighbor amino acid, so each sequence has a slightly different pK, but in principle, most acidic amino acids will be protonated and most phosphate groups will be deprotonated. Many times these enrichment techniques are used in combination. Like I said, many protocols have been published, but this is just the basic principle. The pH that we work with, we can work in this, like here in the illustration, in these tiny tips and put the beads. We can also work in batch in an Eppendorf and shake it. And when we are working with big amounts, some people work in big falcons, or one can do a column containing these beads. All the peptide digest is passed through this column in this case. The phosphopeptides get attached, and the ones that don't are washed. And with basic pH conditions, we can elute our phosphopeptides in the sample. So the other way of enriching is by strong cation exchange. That I don't know if you have heard about this way of separation yesterday in the chromatography, but basically it separates based on charge. So in this case, the phosphopeptides will elute at the very beginning of the gradient, and is a kind of indirect way of enriching a little bit for our phosphopeptides. This is just an example of the group of Steve Gigi and Judith Villen published this paper now some years ago that in the moment was quite a hit to see thousands of phosphopeptides by combining IMAC and strong cation exchange. So they fractionated, and even though at the beginning of the strong cation exchange you get most of the phosphopeptides, then by enriching each one of these fractions, they could see a lot more phosphopeptides. So this is something that is always also to bear in mind. When you have low abundant modification or a low abundant protein in general, by fractionating you always can go deeper. As the instruments get faster and they get better, people also have a tendency to fractionate less, because fractionating complicates the situation then for the data analysis. So now the tendency is going to long gradients, best instruments, etc., and try to avoid this fractionation. But you will always get a little bit more when you fractionate. This is another example of what I was telling you now of the group of Albert Heck. This was published by Shabazz Mohammed, in which they thought, okay, how can I get the most phosphopeptides in our sample? And they did this experiment of, I'm going to do 3 strategies. One that they call the 1D strategy is like, all they did was enrich by these beads, that is a combination of titanium dioxide and IMAC, which now are also quite widely used, and they are actually commercial already. And in this moment they were not commercial. They enrich and they see how much can we get. Well, they were able to see 3,500 phosphopeptides. If we combine this with helix fractionation, we take this enriched sample, we fractionate it, we see how much we can see, and the number of phosphopeptides is a huge increase of like 10 times almost, or 5. And then they wanted to go even further. They said, we're going to do a 3D strategy. So they first did anisotropic cation exchange fractionation. They pulled some of these fractions, they enriched, and they went back to do a helix fractionation. So with this strategy, obviously the numbers always increase, but we always have to balance, how much work is this, how much money this takes to analyze all these samples, and very important, the amount of initial material that we start with. Because the more things we do, the more we fractionate. Here you need to start maybe with 30 milligrams or 100, and here you start in the range of micrograms. So these are all things that we need to take into account when we are going to approach this, not necessarily like an analytical experiment, but a biological question that we want to answer and what we want to know. Okay?
Sorry, a question.
Yes.
The phosphates are stable in the phosphopeptides? Are quite stable, but they support
this is another issue. They are stable up to a point. People like to, in general, enrich and analyze quite fast. More than, it's like how stable is a peptide in the freezer. In general, pretty stable. But phosphopeptides, even less. Phosphopeptides also attach to some of the materials, like, like glass or some metals. So with phosphopeptides, you have to take extra caution, for sure. It's not something very particular that you do, but yes, you encounter more problems like that. So now we have been talking about how we deal a little bit with the sample preparation when we have a phosphopeptide sample. And now we're going to see about which strategies in mass spectrometry we do, because we also need to think about that. I think yesterday you saw some strategies for peptide fragmentation, and some of these are going to be repeated here, but specifically for phosphopeptides. Okay, one of the most common ways of fragmenting peptides is collision-induced dissociation. What happens when we break the peptides this way and we have a phosphopeptide is that in many cases there is only, or is a pretty big loss of the phosphate group. So we end up with our original peptide minus 98 because the phosphate is 80 plus one loss of water, gives this analyte, which is our mass minus 98, which is good, is indicative that we have a phosphopeptide. But what happens is that we don't know, we lose the information of the site of the phosphorylation. And if this signal is the majority signal, what happens also is that we are losing information about the sequence. So if we have this type of spectra, okay, it's great, this peptide was phosphorylated. Where it was phosphorylated, we don't know. And which peptide it was, we don't know. Because we are missing all the other Y and B ions that explain the sequence. So sometimes, this is a problem.
I'm sorry. Can you analyze it in the negative ion mode?
Yes. Phosphopeptides can be also analyzed in negative mode because they have that negative charge. It's not the most common way, because these other strategies that have been developed in mass spectrometry work better. Many years ago, yes, people used to look at phosphopeptides in the positive, in the negative, everything, to get the most of the information. Now it's not very common, although, yeah, it makes sense. So one strategy that is performed quite commonly is this double CID fragmentation, which is called the multi-stage activation. Maybe you have heard about this, MSA or multi-stage activation, if you work with phosphopeptides and they tell you, oh, we have analyzed this with multi-stage activation. What this does is that there is a first fragmentation of CID, which would be the spectra, in which you can see that most of the fragments are the loss of the phosphorylation. And then we tell the machine that every time you see this loss of phosphorylation, take this peak and fragment it again. So then we are gaining the information about the sequence of the peptide. And this is combined in this pseudo spectra, because it's the combination of these two spectra, the one of the neutral loss plus the fragmentation of that neutral loss peak. Okay, and like this, we recover this information. Another very common way of analyzing phosphopeptides in mass spectrometry is by HCD. Why? Because with HCD, in general, we have much nicer peaks and more defined the sequence. And we don't get that big peak of the neutral loss that we get with CID. This is another way of fragmenting peptides that you probably heard also yesterday, that is ETD, that is the electron transfer dissociation. That, as you heard, the fragments that it gives is not the Y and B ions, because it fragments not in the amide bone, but in another one. So the ions that it gives are the Z and the Z. And the advantage of this method is that the phosphorylation is not lost. So we retained that information. However, this way of fragmenting doesn't give always very nice spectra for each peptide. It's very dependent on the mass of the peptide and the charge. So, it's always complementary and it adds information, but there is not one way that solves everything. That's why one of the things that has been implemented to identify phosphopeptides is this decision tree that in the last generation spectrometers is implemented, it can be done routinely, is that we tell the instrument that depending on their mass and their charge to choose one or another way of fragmenting. So online the machine is thinking, okay, this is peptide, this is a mass, this is a charge, I use this method or I use the other. And then we can use all this information to identify our phosphopeptides. More recently, it has also been described, this dual fragmentation method called ETHCD, that combines the fragments of the HCD, or high collision dissociation, with the fragments of the ETD. And we get this double information all together in one spectra. It's like this hybrid spectra. This was published now 2 years ago. And this is now also implemented in the new generation instruments. So you have any question about the peptide fragmentation? Okay, so another of the important points in modifications is to know which is the exact residue that was modified. Okay, so PTM, site localization. We have been talking about peptide scores, Christian told you, and yesterday you also saw that when we do a search in the database, we get a score. What this score tells us is how can we trust that that spectra is that peptide? Okay, great. Here we have one peptide, is phosphorylated, and we have the score of 110. Okay, that's a good score. We believe it. But it doesn't tell us anything about where this phosphorylation was. This peptide has 2 serines, we don't know. What do we do? Okay, if we look at the exact spectrum, these are the ions that we have, okay? Starting from the C-terminus this way comes the Y ions. Here is the Y1, Y2, 3, 4, 5, etc. In blue, we have the ions that are in the spectrum, the ones that we see. And in red, the B ions. In black are the ones that are the theoretical, but we don't see. Okay, in this case, we have this ion, 694, that is telling us that this serine is phosphorylated. It's a beautiful ion here, there is no doubt, it's here. We also see the ion that results from losing the phosphorylation. So there is no doubt. This peptide is phosphorylated and is phosphorylated in this serine. Okay, great. Serine 3 phosphorylated. However, in this case, we also have a peptide with 2 possible serines. And when we see which ions identify this peptide, we have all this Y series until the Y8, which is exactly this alanine. We know all of this, and we know the peptide is phosphorylated, but we don't know if it's in this serine or in this serine. We look at the ions, and there is no ion that can explain that. So in this case, we have an uncertainty. What do we do? Well, a little bit what we were doing now, no? We can do a manual verification, go ion by ion and see. This is, of course, very subjective and depends on the experience and the patience of the person looking at the spectra. Many PTMs have been published with, some doubts about where was that phosphorylation. A closer inspection tells us that some things that have been reported are not so clear. And the good news is that there are also some engine tools that can help us with that, and they can give us a probability. So these are some examples. And in this case, by using the phospho-RS, that is one of these tools to give an automatic probability of the phosphosite, tell us that this serine is 100% probability that is in this serine and 0% in this one, while in this case, the tool tells us that we have 50% probability that is in one or the other. So that, corresponds with our manual inspection, but we don't have to go like this when we have thousands of phosphopeptides. Another thing to take into account is that when we are looking at the phosphorylation in our sample and we see that from here to here we have 3 times more phosphorylation, this can be due to an increase in the phosphorylation per se or an increase in the protein abundance that we had in the sample. Okay, this is, we need, depending when we stimulate, how long we wait to collect the cells, whatever, we also need to keep this in mind. And as an example of phosphorylation in cancer therapy, as you know, kinases are very much involved in driving cells, and sometimes they can also explain the resistance to some drug treatments. So what these people do, this was published 1 year ago, was look at this BRAF mutated cell line of thyroid cancer with these small molecule inhibitors of this MAP kinase pathway, and they were trying to explain why this cell line was resistant to these drugs. So they treated them. They saw that by shutting down this pathway they could see going down all these substrates in their phosphorylations. However, they saw some substrates going up that were not necessarily downstream from this BRAF inhibition, and therefore they postulated that CK2 could be involved. So what they demonstrated is that by inhibiting these both kinases, they could see some synergic effect that explain, and actually the proliferation of these cancer cells decreased. Okay, so with this, we can close the phosphorylation chapter. Do you have any question about phosphorylation?
So for the case of, so this case of site localization, there was no solution because...
No, we not always are able to know where was the phosphorylation. If there is no, that ion, that is the indicative of where the phosphorylation was, we won't know. Of course, we can always use our previous knowledge. Sometimes because we know that the kinase have certain motif, like serine proline. Maybe we don't have the ion, but we know that that's what the kinase is looking for. And then we can use this other knowledge, but by mass spectrometry, we cannot always assign the correct site.
To control for this protein expression, to quantify it, you would do it in the same sample, right? And then it will only be the intensity of the peak that you compare.
Yeah, okay. So what we would do is in parallel, from the same samples and cell lysate, for example, do an enrichment and analyze the sample without enriching, to have, the picture about the proteome of that sample, and in parallel the phosphoproteome. How would we quantify this? In the next session, we talk about quantification. But yeah, we look at intensities, areas, all that. The specifics, we will talk about. But yeah, it's important, to look at the proteome. We would take that same sample without enriching for phosphopeptides. Okay, so now we are going to talk a little bit about glycosylation. I don't know if some of you are interested in this. Glycosylation is one of the most common modifications in mammalian cells. They have a lot of different functional roles, like cell adhesion, recognition of antibodies, or viral infection. They are involved in metastasis in cancer. And they are mostly in the membrane of the cell or in the body fluids. And that's what makes them quite, attractive as biomarkers. Okay, a glycan is actually a sugar attached to a protein. And there are many types of sugars. We have this glucose and mannose, galactose, and not only that, but they also have all kinds of different ramifications. So the world of the glycans is, a world parallel to the proteomics, is the glycomics. And we are not going to go into that today. What we are going to focus is on identifying proteins that are glycosylated. We are not going to look at what was a glycan. Okay, more concretely, glycosylation can be O-linked, meaning that the sugar is linked to the hydroxyl group of the serine or the threonine, or N-linked, that is what we are going to focus on, that is that the sugar, the hydroxyl group of the sugar, binds this amine of the asparagine. Okay? And this is what we are going to talk about today. Like I already said, glycosylation is prevalent in extracellular proteins, secreted proteins, and body fluids. Therefore, they are great candidates as therapeutic targets or, in general, diagnostic biomarkers. Another reason why some people enriching glycoproteins is because it's a way of simplifying our sample. We reduce the complexity. We only look for those proteins that are glycosylated. It's a strategy. So how do we do this enrichment? Okay, this is the sugar attached to our protein. So it can be oxidized in a way that we convert this into a aldehyde that can react with the hydrazide. Okay, this hydrazide is attached to a bit, so now we have our protein, our glycan attached to the bit, and after trypsinizing, where we had the protein, now we have the peptide. This would be the asparagine attached to the sugar, and with the enzyme N-glycosidase F, we cut this bond, converting the asparagine in one aspartic acid. So we have a bunch of peptides with aspartic acid. Each one of these peptides belong to a glycoprotein initially. So now this is easy and ready to be analyzed by mass spectrometry. The same strategy can be used to study glycoproteins that are in the cell surface specifically. This is quite interesting because this is not always easy to work with membranes. These proteins are very hydrophobic, low abundant. By flow cytometry, for example, we cannot multiplex so much, or we don't have the specific antibodies or whatever. So it's the same strategy that I just explained, but in this case, after oxidizing our sample, the aldehyde reacts with this hydrazide that is attached to a biotin. Therefore, we can tag each glycoprotein that we had in the cell surface. And then after lysing the cells and trypsinizing the sample, we end up with a bunch of peptides, some of which are tagged with biotin. Therefore, we can purify them with streptavidin beads. And then again, we had a bunch of peptides. They all have that aspartic acid, and all these peptides were previously belonging to these surface proteins. So this is an example from the lab of Rudy Abersol, in which they were looking for signatures for aggressiveness of prostate cancer, for which they took 4 different types of cells, from normal cells to non-aggressive tumor cells, aggressive tumor cells, and metastasized tumor cells. And they did this strategy of capturing for N-glycoproteins. Why? Because, like I said, these are great candidates for diagnosis markers. And also, they are easy to analyze because we reduce the complexity. So using this strategy, they end up with a couple of markers for PCA aggressiveness.
Sorry, it is possible that during the protein oxidation, either side could bind to other groups, not only to glucose?
Excuse me, can you repeat the question? Is it possible to determine
this reagent, the hydrazide, binds to other oxidized groups?
Yes.
Methionine or I don't know. Not only glucose, oxidized glucose.
No.
This is also very specific for this.
Yes. I mean, it can oxidize a lot of things in the sugar, but it doesn't matter because...
Not only the sugar, in the whole peptide chains.
No. No, it works really well. You really end up with only those peptides, and no, you don't get to see those modified amino acids. Okay. So now we are gonna go to ubiquitination. Okay, ubiquitin is this protein here. It has 7 lysines in the sequence. And as you all know, it plays a very important role in protein degradation. Okay, so a molecule of ubiquitin is conjugated to a protein thanks to these 3 ubiquitinated enzymes. And different molecules of ubiquitin get attached one after the other, creating this change that is recognized by the proteasome. So it's like this ubiquitin is marking the protein that is ready to be degraded. Okay? These are the 3 enzymes that are involved in this process that takes place sequentially in 3 steps. There is an activation, a conjugation, and the ligation. The important thing here for us is to know that this ligation takes place, so one molecule of ubiquitin is conjugated to our substrate protein through a lysine. Okay, and this can be done sequentially, so different molecules of ubiquitin get attached to our protein. And this is the protein that we want to identify, okay, which one has been ubiquitinated. Okay, this is an illustration of what I was just explaining. The 3 ubiquitinated enzymes and the deubiquitinated enzyme. Here we have a monoubiquitin and the polyubiquitin, and all these are linked one to the other by the lysines of the ubiquitin. Okay? So in green you have the structure of the ubiquitin with all the lysines or arginines and the rest of the sequence. This is attached to the lysine of our substrate. And this is just to illustrate that once we have this and we add the trypsin is going to cut after arginines and lysines, and therefore we end up with our peptide that was in our substrate attached to 2 glycines, because it's going to cut after this arginine. Therefore, our signature peptide that tells us that this protein was ubiquitinated is this 114 that corresponds to this gli. Okay, so this is the good news about ubiquitination. We have a signature peptide. But like always, we cannot take our sample complex and now we're going to look for gli in our peptides. We don't see anything, 0. So it's very important, we need to enrich because these are very low abundant peptides. And the way that is used more commonly to enrich for ubiquitination is with antibodies. Okay, there are antibodies created against this gli, and they work pretty well. So again, just what you need to remember is that this is our signature peptide, and this is how we're going to detect these peptides by mass spectrometry. Very recently, the date is July 2nd, 2015, so I guess in 2 days it's published in paper. This molecular cell from the group of Steve Gigi, that lately this group that also we talk about when we were talking about IMAC and has published lots of things about phosphorylation, is lately working a lot in ubiquitination. So for those of you that are interested, this is a nice paper to read about the whole ubiquitination involved in DNA damage. So you have any questions?
But only the lysine can be ubiquitinated?
Sorry, we didn't see you.
Only the lysine can be ubiquitinated? Only the license.
Yes.
The license or the license? Oh.
License.
Lysine, yeah, the
After license.
NH2. The bond is created with the amine group of the lysine. And then the same does the ubiquitin.
But if a lysine, if another amino acid rather than glycine is before the lysine, then you can enrich by this.
Let's see.
You are only purifying, enriching this GG lysine.
Yeah.
And if you have,
If in this same protein you have other lysines,
without
and they are, excuse me?
without the glycines, then you cannot enrich this, right?
The thing is that the ubiquitin always is, this is the ubiquitin, and this is a second ubiquitin. Okay, this is one ubiquitin. So the ubiquitin always, this is, the sequence of the ubiquitin, so always has R, G, G.
It is possible to determine the ubiquitination when you have several lysines, one to the other, so very close lysines.
In your protein,
Yeah.
for example, many lysines together, well, then the problem becomes that the peptide is too short for mass spectrometry. Yeah, this is an added complication, and then one would need to find different strategies. Of course, every protein, every peptide behaves differently, and one needs to study case to case. One thing is to do, like in this case, the group of Gigi, I'm going to look for ubiquitination in general. Okay. The other thing is that you have your protein, you know it gets ubiquitinated, and you know the sequence of your protein, and then you can study particularly. You know, my protein is very difficult because it's full of lysines. Okay, then we need to look at it. We need to think about, this is, all the things you need to know and you need to think about when you're going to approach your exact question.
You could use other enzymes,
Exactly.
but then it also, your side chain also changes.
Exactly. Exactly, yeah. Just as if you have a protein full of lysines, ubiquitinated or not ubiquitinated, you have a problem. Okay. So I think we're gonna spend now 10 minutes max talking about methylation and acetylation, particularly in the case of histones. Okay, histones, you all know that play a very important role in gene regulation. They are these molecules that assemble in octamers, forming the nucleosomes that they wrap the DNA, giving it a structure and forming the chromatin. Histones have these N-terminal tails that protrude from the nucleosome, and they are highly modified, specifically with acetylation and methylation. That's why we chose this example to talk about these modifications. And this specific modification, that is what has been called the histone code, the way they are modified, it can interfere in transcription, DNA repair, and chromosome structure, etc. Okay, so we are going to focus on the acetylation and the methylation. Okay, for the methylation, methyltransferase adds one CH3 group in a lysine or an arginine. So one of these hydrogens gets substituted by a CH3 group. So we have in our peptide a plus 14 Dalton. 2 units can be added, or 3. So we have monomethylation, dimethylation, or 3-methylation with different masses in each case. And in the case of acetylation, we have an acetyltransferase that adds this group, COCH3, that has a mass actually quite similar to the trimethylation. Okay, these are the masses that are added to our peptides. This is what we need to know for mass spectrometry. Okay, this in the old days used to be a problem. These days with the mass accuracy, which if you remember, mass accuracy is how exact is our mass, because now instruments nowadays are quite accurate in the mass that they measure. We can distinguish these modifications, the trimethylation from the acetylation. There are also some diagnostic ions, like we had with phosphorylation, some neutral losses, some ammonium ions that can give us keys about having, these modifications. And then, like with all the ones that we have been discussing, fractionation can help and enrichment with antibodies. Okay, this is more or less how we deal with these modifications. The histone case is, a little bit more complicated than this. Why? Because like you, Victor, were saying, there are loads of lysines and arginines in our protein. Okay, these are quite small. They are very hydrophilic. And they are very hydrophilic because they have all these lysines and arginines. So what happens is that when we use trypsin, the peptides are tiny and they are not good for our mass spectrometer. And on top of that, we want to see all these modifications. So one strategy that has been used a lot, that has been, the group of Benjamin Garcia has been working a lot with this, is derivatization, which is alkylating these lysines with propionyl. They use propionyl anhydride to alkylate the sequence. So when we have one methylation, for example here, it gets also propynylated in the lysine. Same for the lysines that are not modified. And those that are already full, these lysines that already has the 3 methyls or the acetyl, they don't get modified. So all we are trying to do here is that when we cut with our trypsin, we get fragments of peptides of the right size and that are good for our mass spectrometer. And all that we need to take into account is that then when we go to the database search, we need to tell that, be careful, lysines have this propionyl group in them. Okay, so by adding this in our database search, we will be able to find these peptides. Yeah?
But you will still have many peptides, no? With the peptides will be very small.
No, but this is now the right size. Instead of having here this of only 4, this of only 3, this would be, all these would be too small. Now we have them double size. And just, because of the time, I'm not gonna go through this paper, but you can look at it. Just to mention, they studied how the information of these PTMs is transferred within a cell cycle, which is very important for epigenetics, how this histone code is propagated, and they, by following specific unique peptides, those that only belong to one specific histone and their modified counterparts, they could see how these modifications change across one cell cycle, and how within one cycle the modifications get restored from the parent ones.
Just a question that it's really outside of the topic. It's nowadays can they link this modification in the histones with the gene that is associated with this histone or not?
I'm not sure if I understand your question.
Yes, you know that the histone modifications,
Mhm.
they are related with epigenetics and
Yes.
the translation.
Yes.
So are there any techniques linking what we see in mass spec?
There are studies on proteogenetics that try to look for these things. I don't know how, I mean, not the whole thing is well understood, but yeah, there are a lot of studies and a lot of interest in this, for sure.